Hello and welcome back. We're going to continue talking a little bit about protein and protein structure. We talked last time about the monomers that make up proteins, and those monomers are the amino acids, and those amino acids are linked together to make longer polymers. Okay? It's the longer polymers that make up our proteins. Okay? Our monomer is the amino acid. And because we're building longer chains, because we're building polymers, those different functional groups attached to each amino acid are going to impart different characteristics onto the protein, all right? And in terms of that, the protein is going to be organized in a uniform way. And my pink's kind of running out, so we'll switch to yellow, okay? So in terms of our level of organization, we're going to have the primary structure of the protein. We are going to have the secondary structure. We're going to have tertiary structure. And we're going to have quaternary structure. All right? So in terms of our primary structure, we've got the order of those amino acids. So when I drew the two amino acids in the last video, whatever those R groups are would dictate the order of the particular amino acid. So in terms of primary structure, we might have a leucine, a glycine, a tyrosine, okay? Those are names of one of the 20 amino acids. So the order that they're put together is going to be the primary structure. And when I have that leucine, glycine, tyrosine there, what that's referring to, if I draw our amino acid up here, say this functional group down here is the functional group for the leucine. Then we've got our next amino acid, with our functional group here being the glycine. Again, I'm not drawing the functional groups because I don't care if you know the actual chemical structure of the functional groups. And I'm just using some of these as examples, okay, to show you how this is going to work. Then we'd have another nitrogen and an amino group, another carbon, hydrogen, and this last one I gave was tyrosine. Okay, so that would be how our primary structure is. Typically when that's drawn out in textbooks, it's drawn linearly. Your textbook draws them as circles. That's fine, okay? But essentially that's the primary structure. It's the order of the amino acids, all right? Now each of those functional groups, because it has a particular chemical structure, are going to be able to interact with other functional groups in this primary sequence, okay? So in terms of that secondary structure, now with secondary structure, we're talking about interactions that happen, chemical interactions. And primarily the chemical interaction that we're going to talk about is going to be hydrogen bonding. Okay? Because what's going to happen, secondary structure, I always kind of draw this as a pig's tail. You can have some hydrogen bonds that occur between the functional groups that result in essentially a coil being generated. And that coil we refer to as an alpha helix. Okay? You can also have hydrogen bonding happening between different strands, and I'll draw those hydrogen bonds as dots. In this case, there you'd have hydrogen bonding kind of in that area. Okay? So these are our hydrogen bonds. These are our hydrogen bonds. And when they're zigzags like this, it's referred to as a beta pleated sheet. You may also see it referred to as a beta sheet. That's fine, either's fine. But essentially what it is it's those first sets of chemical interactions that are going to happen because of the functional groups that make up the primary structure. So whether you have an alpha helix or whether you've got a beta pleated sheet, all right, is going to depend on what the primary sequence is. But the secondary structure associated with protein are going to be the alpha helix and the beta pleated sheet. Okay? Our tertiary structure now is going to be the overall structure of our protein. So in terms of that, we may have an alpha helix, we may have a beta pleated sheet, we've got an alpha helix, maybe we've got another beta pleated sheet and a beta pleated sheet, okay? So really the tertiary structure is going to translate to the three-dimensional structure of the protein. Because the hard thing with the textbook and with me drawing this on the piece of glass here is it's very two-dimensional. But proteins have a physical shape, just like your coffee cup has a physical shape, all right? And your coffee cup has an opening where you put the coffee. Proteins are going to have regions of the proteins that are going to be important in how it functions. Now, if you take your coffee cup and it gets dropped on the floor, shatters, you no longer have that three-dimensional structure that allows you to hold the coffee within the cup. The same is going to be true with proteins, okay? If you disrupt how they're put together in space, in 3 dimensions, they're not going to function, okay? And in terms of that, if we disrupt the 3-dimensional shape, what we refer that to, the scientific term for that, is we say that protein has been denatured, okay? And if you want an example of something where day-to-day life you may denature proteins, all right? If you made an egg for breakfast this morning, all right, and say you wanted to go over easy on your egg, you crack that egg, you put it in the hot pan, that egg, the outer part, goes from being clear to white. The heat from your stove has denatured the protein in that portion of the egg and has converted it essentially into a different shape, okay? So that's kind of an example of how a protein can become denatured. And in terms of denaturing proteins, all right, there are a couple ways that we can denature proteins. Heat being one of them, all right? So if you cook your egg on the stove, that's going to denature it. pH changes can also denature protein. So if the environment becomes too acidic or if the environment becomes too basic, that protein isn't going to function, because essentially what happens when you're talking about denaturing a protein, you're disrupting the hydrogen bonding that's happening, okay? And in terms of that, you may have some hydrogen bonding and some other bonding that's happening in your three-dimensional structure as well, or your tertiary structure as well, okay? So heat, pH changes, salt can also interrupt the three-dimensional shape, okay? Because the ions essentially pull things apart so they aren't in the exact same shape that they were before, okay? So in terms of our proteins structure and our organization, we've got our primary, we've got our secondary, we've got our tertiary here, all right? The quaternary I'm going to talk about. We're not going to focus a whole lot on quaternary structure in this class, but when you get to A and P, you're going to have proteins that are going to have quaternary structure, all right? And what it means for that protein to be quaternary, this is going to be protein 1. Do that in yellow. Let's go with pink. This is going to be protein number 2. So what you have with quaternary structure is you have multiple chains, polypeptide chains. So you have multiple primary sequences that are held together, all right? Great example when you get to anatomy and physiology. Hemoglobin is put together in quaternary structure. There are actually 4 peptides that compose hemoglobin, and those 4 peptides are going to be important in how that hemoglobin functions. For now, I'm not real concerned that you know how hemoglobin functions, but know that hemoglobin is an example of a protein that has quaternary structure. Quaternary, or our 4th level of organization, just means you have multiple protein chains linked together in order for that to be a functional protein, okay? The majority of proteins within your body are going to function based on their tertiary structure, okay? Now in terms of that, if you have a quaternary protein, that quaternary protein still has tertiary structure, it still has secondary structure, it still has primary structure, okay? So don't think, try not to confuse that, okay? But in the case of proteins, why this is important, okay? The three-dimensional shape, and I spend most of my time talking about tertiary structure because the three-dimensional shape is important, okay? The three-dimensional shape is going to be what determines the function. And really what we're going to discuss mostly in this class when we get to chapter 5, we're going to talk specifically about enzymes and the three-dimensional shape associated with enzymes and why that's important, okay? So for now, what I want you to know, the levels of organization associated with proteins. You've got primary, secondary, tertiary, quaternary, and what the difference is between each of them. Right? If you have any questions, please let me know. Thank you for your attention.